Two announcements. One of them you're going to like very much. I'm going to postpone the deadline for homework 3, not 2 is passed already. So assignment 3, I'm going to postpone it 6 days to 15th of May, okay? The reason is that we are going to have time until May 27, and for your last homework, I'm going to give you 12 days for your last homework between May 15 to May 27. The last days of classes are May 27. So 12 days for last homework is going to be enough for you. It's going to be much easier than the first 3. I mean, the first one was easy, but the second and third one was difficult homeworks. The last one is going to be an easier one. It's going to be about protein structures. And 12 days are going to be enough for you to work on this homework. You are going to be working with 3-dimensional data, but you are not going to be producing any 3-dimensional output. Just some numbers. I have some idea about the last homework. I'm going to announce it after the deadline is over. So 6 days postponed. Homework 2 is deadline 6 days postponed to May 15. And May 15, you are going to get your last homework. Another announcement is your first homeworks are graded. Probably your teaching assistant is already posted on the call, or if not, he's doing it right now. After the class, you're probably going to see your grades. The average was 63. I don't know if it's including non-submissions or not, and there are a couple of, there are lots of hundreds, full grades. There are partial grades, between 55, 75. So it's average. The performance is not very bad, but considering the homework was not very difficult, that's not a very good average. I mean, 63, I was expecting more, but probably it may include the zeros, no submissions as well, because there were like only 25% of the class did not submit anything for first homework. So these are the 2 announcements. I already posted the solutions to homework 2. I mean, the family profile HMMs for the 4 families 2 days ago. It was 2 days ago. At night, so you can, and you'll have 6 more days to work on this assignment. You are going to use those families, you are going to use the test sequences, you are going to evaluate them with respect to each other and find which test sequence belongs to which one of these 4 families. Okay. Today we are going to talk about protein structure prediction. I forgot to bring my toy. I had this. I was going to talk about the ab initio methods. That toy is really good at showing the ab initio techniques, how it works. But again, I'm going to use one of these cables if I have. Yeah, I have this. I'll stick with this cable instead. So today we are going to talk about structure prediction techniques. The structure prediction algorithms, we are not going to go into details of these algorithms, so you will not be able to implement these algorithms, but you are going to know the general trends, their general strategies, what kind of strategies each different approach use when predicting the structure of a given protein sequence. So we are still waiting for the PC to turn on, but I can start talking about the structure prediction techniques right away. So there are 3 types of main approaches, general approaches to predict, given a protein sequence, the amino acid sequence of a protein, what structure it's going to be in. These 3 approaches are ab initio methods. Ab initio is Latin for from scratch, something like this, from initial, without using any additional information. There are ab initio methods. There is homology modeling techniques, And the final is threading. Ab initio methods do not assume, so by the way, our PC is turned on, so I can start talking about it on the slides. It's here. Yeah. So there are these three main approaches: ab initio techniques, homology modeling, and protein threading. Ab initio technique do not assume any prior information and can predict the structure of any protein sequence. But the bad news is that it's computationally very expensive, and therefore it cannot find structures of long proteins. It's a very computationally expensive technique. Homology modeling makes use of existing knowledge. It uses sequence-sequence comparison. In other words, if we want to find the structure of this new protein sequence, we first look for existing databases, and whether we try to find, is there a similar sequence to this sequence I'm trying to predict? I mean, this is a sequence I try to predict the structure. If there is a similar sequence to this sequence, their structures may also be similar. So homology modeling is based on this fact. If I have another sequence with known structure, and if the similar sequence with known structure, I mean similar to this query sequence, its predicted structure can also be similar to the structure of this homolog protein. So we can make use of this fact, and homology modeling is based on this. If there is a similar sequence to your query sequence, use the structure of this similar sequence to predict your structure. If there is no such thing, I mean, if your sequence is completely novel, there is no sequence similarity to any existing known proteins with known structure, we can use protein threading. And protein threading is a really nice technique which aligns a sequence against a structure. We haven't seen such a thing yet, but it's like this. So we have a sequence, but what structure we are going to compare it with? What threading does is, it uses structure templates. The main idea of threading is, in nature, there are not many different shapes that proteins take. The shapes of proteins are like 1,000, 2,000 different shapes that we observe in proteins. For example, there are long proteins, there are ball-like proteins, there are key-like proteins, V-shaped proteins. So there are these shapes that the proteins can take. There are only a couple of thousand, maybe 1,000, 2,000. So that's the main assumption behind protein threading. We only observe a certain set of shapes of proteins in nature, and if we take from these different, from all the proteins in nature, we take representatives of these different shapes. What we do is we produce ourselves a template, structured template database. For example, there are shapes that looks like this projector, there are shapes that looks like this monitor, there are shapes that looks like the staple, okay? So what we do is, if I have these three shapes, now I ask this question. I have my sequence, I have my protein sequence. Which of these three shapes, so this is my template database, the monitor, the projector, and the staple, this sequence, which of these three shapes is more likely to be the shape of this sequence? So what we do is, we thread this, in other words, we align this sequence on top of these structures. For example, we just take this sequence, if the sequence is going to have this projector shape, I just thread, this is called threading, I thread this sequence on top of this shape and see whether this sequence actually can get this shape. So we have certain energy functions. For example, there may not be a unique threading. For example, this is one way to thread. Maybe what you can do is, okay, I'm going to have a gap here. Maybe another way to thread is this. Another way to thread is start from here, do this. Maybe, using there are multiple ways. Actually, this is also an MP hard problem, unfortunately, to find the best way to thread the sequence on top of a structure, which is going to give you the minimum energy. So, in other words, after we thread this, we look at which amino acids become neighbors on the structure and whether these amino acids want to become neighbors. And we do this thing for other shapes as well. For example, maybe onto this shape, this sequence is going to be more likely, the amino acids are going to be more likely to be closer to each other, like this, if they form this shape. And if they form this shape in this monitor, for example, some amino acids are going to be far from each other. Maybe they like that better. So based on amino acid types, we can find out which structure is more likely for this sequence. So that's the main idea of threading. We are going to do this for all the template structures in our database. We are going to try to find out if the sequence had that shape, what is the energy of that shape? Is it a likely shape for this sequence? This is what threading does. And at the end, out of these template structures, we are going to pick the one with minimum energy, which is going to be the most likely shape this sequence can take. That's the main idea of threading, and I'm going to show you some more examples about threading. So, in ab initio techniques, we use certain abstractions, certain assumptions to reduce the search space. One of them is called the simple lattice models. This assumption, this abstraction limits the search space by assuming that each amino acid can take a position on an integer coordinate space, for example, on a fixed grid, on a regular grid, square grid. We may have a square grid with cells in this grid, and each amino acid can take a position in this grid, and neighboring amino acids on the sequence should be on neighboring grids. What we are going to do is we are going to position this sequence inside this cubic grid, and each amino acid is going to have a certain, its position it cannot be in a floating point number. It's going to be in a simple integer coordinate space. And what we do is we also abstract the whole protein sequence. A higher abstraction is just two types of amino acids, hydrophobic and hydrophilic, or hydrophobic or polar amino acids. In other words, so we are these 20 different amino acid letters, they are going to be reduced to 2 letters only. By looking at the amino acid physiochemical properties, we are going to see whether this amino acid is polar or not, whether it loves to be near water, or whether it's hydrophobic, it doesn't want to be near water. We are going to just use this physiochemical property to abstract the sequence into a sequence like this. Our protein sequence is going to be a sequence like this: P, H, H, P, P, H, P, 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 H, something like this. We are going to convert our amino acid sequence into a letters of polar or hydrophobic amino acids. And the only force here would be, our goal will be to position this sequence of amino acids on the grid so that hydrophobic amino acids are going to be in contact with each other. They are going to hydrophobic collapse. That's the force, which is the hydrophobic collapse, is going to pull together these hydrophobic amino acids, and the polar residues are going to try to stay outside. And it's like a puzzle. For example, if I use a 2D grid, here's a puzzle for you. Okay, this is a 2D grid. Position this sequence onto this grid, so you're allowed to do some things like this. So I have this P, H, H, P, P, so you can do something like this. P, H, H, P, P, H, P, 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 H. Okay, so it goes like this. So this was my sequence, and I positioned it on the grid like this. So at each, you can use, for example, from each grid position, you can either, for example, here I was going this way and I turned right. And here I was going straight, I turned left. You can think of these as you're threading, again, you're putting a string in this, placing a string on this grid, and you are allowed to make moves, either go straight at each junction, or go left, or go right. Okay? And here's a puzzle for you. Position this sequence onto this grid so that the number of hydrophobic contacts are maximized. So what's a hydrophobic contact? This one, for example, has 0 hydrophobic contact. For example, if I had this, if at that point, let me try to, so I have P and an H, yeah. If I had 2 P's, okay. P, it's going to be difficult to have this. This is a difficult example, but I assume this is also a contact. Usually the diagonal neighbors are not considered neighbors, but if 2 hydrophobic residues that are far away in sequence are neighbors on this grid, you get a score of 1. Okay? Do you understand hydrophobic contact? So here, and this is not a hydrophobic contact, because no matter what you choose here, they're always going to be neighbors, because they are neighbors in sequence. These 2 H's are neighbors in sequence, so in all the solutions you have, they are going to be neighbors. So you don't score, there's no need to score them. What we score is these far away. For example, if you can make this in contact with these 2, you are going to score 2. Each of these contacts, which are far away in sequence, if you make these hydrophobic amino acids come close to each other on this grid, you are going to score yourself 1 point. So the problem is, what is the conformation that gives you the maximum score? So how do you, given the sequence, how do you maximize this? So here is one try at this, at the solution. Let's try to, okay, let me erase all these parts. So I'm going to have P, H, H, and I have P, P, and H. P, P, H. So I went this way. As you can see, I scored myself 1 point at this point by making these 2 close to each other. Again, it says to be specified whether diagonal neighbors are going to be considered as neighbors or not. It has to be given in, it's a parameter that is specified, but usually it's not considered as a contact, so I'm not going to consider this as a contact, so I'm not giving me many points because of this. I got myself 1 point because of this contact, and what I have is the remaining P, 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 H, 3 P's and H. What can I do to, maybe I can do this. How can I make it, the last one, in contact with something. Here, P, where am I going to? H. So this is not considered a contact, so it's not going to score anything. Okay, I changed my sequence, make it 2 Ps. You're not allowed to do it in general. You cannot change your protein sequence when you're predicting it, but just to show you how we can get additional contacts. So HPPH, that would give you 1, 2 score, so it's going to give you a total score of 3. Okay, we only had 4 hydrophobic residues and we get a, so as you can see, we got what we have ourselves. This perfectly shows, this actually perfectly shows the polar residues are on the surface and the hydrophobics are at the core. They are in close contact to each other. Our protein structure is going to be most likely this. This is how we predict it, okay? It's a score of 3. But as you can see, yes? So one is, so our sequence is like this. Let me use a different color pen. So this is our sequence. Okay, this is our protein sequence. Here I got 1 from these 2 H's, and I got 1 from this one and 1 from this one. The first one, they're already neighbors because they're close in sequence. So I do not score contacts that are already neighbors in sequence. As I said, it's just an option. I mean, it should be specified in question. I mean, diagonals may be considered as neighbors or not. And even I may specify something like this. I may allow additional interesting moves instead of just left, straight, left, right. I may also go 45 degrees to right. I may go to diagonal neighbors during action as well. For example, from here, you can jump here or go back here. So this is just, it's going to increase your solution space, but this is how much obstruction you are going to have. This is the definition of the problem. But even this simplified problem with just 3 actions at each step, your solution space is huge. Okay, I mean, therefore, even the optimizing, maximizing the number of HH contacts is a very difficult problem. There is no efficient, known solution to solve this puzzle. I mean, this was only a protein of it like 8 or 9 amino acids. Consider you are doing this for 100 amino acids on a 3-dimensional grid. The number of different shapes you can have is enormous. I mean, the solution space is huge. Ab initio techniques try to exhaustively search these solution space, maybe by using some intelligent strategies. They try to optimize these hydrophobic contacts, but solving this lattice model is very difficult. And this is just to show you, and by solving the structure, we didn't use any additional information other than sequence. We had the sequence, and we just wanted to put the sequence into minimum energy type of form to predict this structure. That's why it's called ab initio models. We didn't use any information about any other sequence, any other known structure. We didn't use anything. We just used physics, the hydrophobic properties. And we just obstructed our coordinate space by a grid structure. So the puzzle, the toy that I was going to bring here was something like this. It's actually a cube, like Rubik's cube. It's a cube, but the cube you can just disentangle the cube. It's a linear string of smaller cubes that are attached to each other by strings. So you can, if you dismantle this cube, you have a linear smaller cubes. And the puzzle, this toy, your goal is to bring all these cubes together to form this one large cube. You cannot perform every action. These strings are attached to each other in a certain way. And you can only perform certain actions between certain smaller cubes, and it's very difficult to solve this puzzle actually. I mean, to make it a cube. So it's similar to this protein folding problem. That final cube shape may be the one which maximizes this hydrophobic context. All the other ones has larger volume. It's going to occupy larger space, and you can have millions of different shapes between these strings of smaller cubes. You can have, I mean, not millions maybe. I mean, I cannot pronounce the number. The solution space is so huge that you cannot try an exhaustive search of this space. So here's another example which scores 5. And the 5 comes from these dotted lines show our scores. As you can see, this is not scored because they're already neighbors. The brown ones, or the browns one are here hydrophobic amino acids, and these are the long-range interactions between hydrophobic amino acids. It scores 5. And sometimes our score, we may also use different type of scoring schemes. For example, if we have a buried polar residue, in other words, if we treat the boundary of this shape as the molecular surface, for example, this polar residue here, actually these two of them, it's like they are in the middle, they are stuck in the middle of this surface. If I'm considering a 2D surface, they are going to be inside the molecule. You can penalize this in your solution. If you want all the polars to be on the surface, you can penalize either buried polar residues like this, or all these 4 hydrophobic, all of them are going to be on the surface. You can also penalize this. You may not want hydrophobic residues on the surface of a molecule. And in general, our molecular structure is 3-dimensional. So this just shows a 2D lattice, but we are going to have a 3-dimensional grid structure, like a cubic grid like this. We are going to place these amino acids on this 3-dimensional cube. And yeah, the problem is very difficult. No known efficient solution exists to, given a scoring function, maximize the hydrophobic contact. Even this simpler function with optimization function, there is no known efficient solution to find the best shape. So for smaller proteins, we can use exhaustive search. And for larger proteins, there are other optimization and search methods which are heuristics, and most of them are heuristics, which does not guarantee the optimal solution. I'm going to skip this. It just shows that how do we represent such a shape on the grid. We can represent it by using directions. So go forward, turn right, go forward. So we can use absolute or relative directions. Also, we should not allow certain moves, should not be allowed. We cannot allow 2 amino acids occupy the same grid location. So if we are here, for example, we are not allowed to do a right turn. We can either go straight or go left. These crashes should be, bumps should be disallowed. There are more realistic models than this 90-degree grid. There are 45-degree lattice models. At each junction, you can do multiple moves. You can go to diagonals. And there are different search strategies on this, but still, right now, all ab initio models, they are limited. They cannot solve very large protein structures, and it takes a lot of time. They're computationally very expensive. Ab initio models are used rarely, and it's difficult to find the correct protein structure by exhaustive search. We can also use different energy functions to describe the protein structure. The bond energy, the bond angle energy. So if we have a structure like this, we can talk about whether this structure is feasible or not. We can compute by using chemistry and physics. We can actually compute these energies, and nature usually favors a low energy conformation shape. Okay, so these are the difficulties, main difficulties in ab initio methods. There are many degrees of freedom at each step for each amino acid, so computationally too expensive for realistic size proteins. And there are what makes the problem NP complete is the remote interactions, these hydrophobic or polar hydrophobic interactions that are far away in the sequence. When you bring them together, they become interacting, and a local choice may affect a global solution. So that's why the problem is difficult. You cannot use dynamic programming approach or divide and conquer approaches. What they assume is that if you locally find a good solution, this solution is going to remain to be good in the optimal final solution. That's how divide and conquer algorithms work. They divide the problem into smaller problems. However, the difficulty here is that when you solve something locally, when you put it together in the big solution may be bad just because of this. These local solutions may affect the whole solution. So you cannot divide and conquer this problem. It's not possible. And that's the main difficulty in such most NP complete problems. And here, the nature does not do an exhaustive search in all these shape space. Now in nature, there's a folding pathway the nature takes, and in nature, these amino acid sequences, they go to the correct shape, but just very fast. Taking into account these folding pathways is right now, there's research on how this folding occurs. And if we have more information about these folding pathways, we can incorporate into these algorithms to guide our search. And another thing is this is a real problem in other prediction techniques as well, which just shows that the last bullet here says that the structure, the final structure, may not only be determined by the sequence itself. There may be external factors, external proteins, these are called other enzymes or chaperone proteins, helper proteins, which will help this protein to take its final shape. And if you do not take into account these, if you do not know any information about these external factors, we may end up with a wrong prediction. So this is the final bullet is also the difficulty in other approaches as well. So that was ab initio modeling. Now let's talk about homology modeling. Here are the main steps of homology modeling. First, we need to find ourselves homolog or putative homolog sequences with at least sequence similarity of 30% is required. And these homolog sequences, they should have known structures. So what we do is, we get our query protein sequence, we do a sequence similarity search to the proteins in the PDB, protein data bank. There are 60,000 proteins. We hope to find out of these 60,000 proteins, a couple of them have similar sequences to our query sequence. That's what we hope. This is the first step. If you cannot find any such homolog sequence, it means that you cannot use homology modeling. So that's the first requirement. You need to have similar sequences with known structures to your sequence to be modeled. So by using tools like BLAST or FASTA, and we can use BLAST because BLAST allows you, remember, when you do a BLAST search, you can choose from a number of databases to search in. And one of these databases is Protein Data Bank. So you can go to BLAST website and say, input your query sequence and choose as the database to search in. You can choose Protein Data Bank, which is Protein Data Bank is the database of proteins with known structures. So if you get a hit, then you're lucky, means that you have found a similar sequence to your query sequence with known structure. And this is the first step. If you do not get any hit, you are not going to be using homology modeling. And we align the target sequence with the template proteins. So this is our target sequence is our query sequence. If these template proteins are the similar sequence proteins with known structure, we can use multiple alignments. So we align these sequences to each other to show which parts are similar. They may not be completely, they may not be 90% similar to our sequence. So we first identify which parts of these sequences are similar. Maybe if we if we find certain parts from certain sequences, we are going to grab certain structure from one sequence, another structural substructure from another sequence, and we are going to combine them at the end to model our final structure. So we can use cluster W or other alignment techniques to identify conserved regions. So these regions are conserved. Identifying conserved regions is going to help us. These regions are going to probably have that shape. We are going to be able to identify the conserved shapes. And then after this, we build a model of the backbone as modeled first, taking the backbone of the template structures, conserved regions into account. And then the backbone just gives us a rough idea about the protein shape. So the backbone is going to look something like this, for example. And then we model these regions may have different shape. For example, the backbone is like this, but this amino acid here may be pointing this way or pointing this way. So it may have different orientations as well. The side chains may have different orientations. And we work on the details of the structure, the side chains, and I mean the overall shape stays the same, but we work around the other parts of the structure to model the loops and maybe in regions with gaps use other procedures to substitute segments of appropriate length. And side chains are also added to the model backbone, so loops and side chains are different. There may be certain loops are like this. After we find the backbone, we may have found a helix here, another helix here, but this region here, we do not know what it is, and we may not know the actual shape. This part may be here, may be like this, it may be like this, maybe it may be like this. So all of these may be possible. This is the loop modeling part, and side chains is the one I talked about. They may be in different orientation. And at the end, after we get ourselves a structure, we evaluate it, whether there are certain things that are physically incorrect in this model. For example, there may be two atoms that are very close to each other, which is not possible in nature. Maybe you have to observe certain distance between atoms. The bond lengths should obey certain rules in nature. So we evaluate our model and check whether it's a feasible, feasible physical model. There are tools for that, like what check, what if. There are tools named like this, which can get a protein structure and tells you, it debugs this structure. Basically finds errors in this structure, this side chains in incorrect position, and you do fine-tuning on this final model to resolve these incorrect parts. And the entire structure is optimized at the end, and this is what how homology modeling works. And in summary, I mean, we get similar sequences with known structures, and we make use of the structure information in them to model our protein. And there are certain servers that help you. You can use Civis model, and there's a free, what is called modeler. If you want to learn more about this, in the molecular biology department, Professor Çağdaş Son actually has a course on protein structures. He talks about this structural modeling. He talks about this is a free tool. Modeler is a free tool that you can use. He talks more detail about these structural modeling steps. And the last thing I'm going to talk about is threading. As I said, if we cannot find a similar sequence, if we cannot use homology modeling, we can still use protein threading. We don't need similar sequences. Given a target sequence, we can always use protein threading to predict the structure. But it has certain assumptions. The main assumption is that the number of different shapes in nature is limited. The number of sequences may be very diverse, but structure is more conserved in proteins. I mean, you may have different sequences, 30% sequence similarity, but their shapes are going to be similar. So if you look at all the shapes in nature, all the proteins in nature, you are going to observe certain similarities between them. And if you categorize them into, by looking at their overall, for example, this is what we call a tin barrel protein. This is, by its shapes, it looks like a barrel. Here, right now, currently, we know about 700 different shapes, different folds of proteins, and it's expected in many papers, this number is expected to be between 1,000 and 10,000. And 10,000 may be a very overestimate of this. The more accurate estimates are maybe 2,000, 3,000. But in nature, we do not, even if we had known all the structures of all the known protein sequences, okay? So we have 7 million, 10 million protein sequences. If all of their structures were known, we wouldn't have, we would still have this many 1,000 or 10,000, between 10,000 different shapes. Okay, that's the main assumption. And the current protein structure initiative, I mean, there's a lot of meetings by researchers of experimental structure determination scientists. What they do is, as a scientist, what you want to do is, you want to, if you're going to solve, for example, if you have X-ray crystallography machine, okay, nobody is using it right now, okay, it's staying right now, and your goal is to find the structure of a new protein sequence. Which protein? So you have 7 million protein sequences to choose from. Okay, imagine you are this guy. Okay, you have a machine that you can solve the structure of a protein sequence. Now the question is, which protein sequence to choose to find the structure? Okay, here an intuitive thing to do is, if you can find a sequence out of these 7 million protein sequences, if you pick pick one sequence which is going to have a very different structure, a very different shape than all the existing shapes, what you do is you contribute really good to this, you contribute more to science, right? I mean, imagine this, you pick the sequence, you find a structure, and you find that there's already a very known similar structure to this sequence. I mean, you have worked 1 year on solving this protein structure. At the end, you come up with a very similar structure already existing in the structure database. Your contribution would be less. So there are also papers, how can we predict, without actually even knowing the structures, how can you pick a sequence which is more likely to have a different structure at the end? Okay, this is another research problem. So you don't know, this is different than structure prediction. You don't need to predict the structure. Out of these 7 million proteins, pick the sequence which is going to most likely have the different structure than existing known structures. So this is, and right now the structure finding initiatives, they are trying to select such protein sequences. So if we have these machines that can find these, solve these structures, then we need to direct our research, our efforts to these such sequences. Our goal is to increase this, in other words, it's like this. If this is the protein structure space, this is the structure space, these are the known structural shapes right now in the structure space. There are still certain shapes we do not know their structures in this. These are the ones that we know. These are unknown. And our goal is to increase this known structure each time we solve a new structure. Right? Each time we're finding a new structure experimentally, our goal is to cover the whole protein structural universe. And because if we can cover all of them, then threading methods are going to work best. Right now, we have 700 structures, and threading assumes that when you're solving when you're predicting the structure of a new sequence, this is your sequence, threading assumes that the sequence, threading assumes that we know the known part of the protein structure universe is large enough. Okay, it covers maybe 70% of the whole protein structural universe. this known part is large enough that this sequence is going to take the shape of a known structure. This is what threading is based upon. If this sequence here, it's like the reverse problem of the previous problem I stated, how to select a sequence which is going to have a different shape. Threading assumes the reverse. Threading says that if you give me a sequence, first I assume that if I'm going to predict it correctly, I assume that the final shape it's going to take is similar to already known structure. It's in this known part. This is what threading assumes. If it's not the case, then you will not be able to you cannot predict the structure by using threading if it's not the case. If the sequence is going to have a completely new shape, threading is not going to produce you a good answer in that regard. Threading assumes your query target sequence is going to, at the end, have a conformation, have a shape similar to already known structural templates. So let's stop here. I'm going to talk about threading more at the beginning of our lecture tomorrow, and we are going to talk about structural alignment, comparing two different protein structures, and we are going to talk about details of some algorithms to solve that problem, and that's going to be we are going to conclude protein structure after that.